Welcome all. Stay tuned with Modern Group of Institutions Indore. I'm Mitesh Sachdeva, working as an assistant professor in Modern Institute of Pharmaceutical Sciences. In this video lecture series, we will start with a new category that is H2 antagonistic drugs, that is histamine H2 receptor antagonistic drugs. Under this, we are taking a first important drug that is cimetidine, and we are going to discuss about the introduction and synthesis of cimetidine. It inhibits cytochrome P450 along with blocking the H2 receptor. They also inhibit cytochrome P450, and thus it leads to many drug-drug interactions. If given more than 1 month, they may cause certain adverse effects, that is anti-androgenic actions and gynecomastia. This type cimetidine has a good bioavailability, 63 to 78%. Since the drugs which comes under the category of H2 antagonists, they are also used for treatment of ulcers, peptide ulcers or stomach ulcers or gastrointestinal ulcers. Hence, they are also called as anti-ulcer drugs. Apart from this, they are also used for heartburn, Zollinger-Ellison syndrome, and GERD. If we see the mode of action of these drugs, we will find that the drugs cimetidine, famotidine, and ranitidine of category H2 antagonist blocks histamine. They inhibit the histamine to bind with H2 receptor, and as the histamine binding with H2 receptor is stopped, there is no formation of acid inside the stomach. Basically, what happens, the receptor H2, there is a receptor H2 which is present on the post lumen of stomach and in the posterior cells. And when the histamine, which is biochemically synthesized in the human body it is a biochemical reaction, this histamine goes and binds with the receptor H2. It stimulates proton pump. So proton pump is stimulated, and which is also called as H positive K positive ATPase pump. Through the stimulation of proton pump, the pump opens, and the inside hydrogen molecules, hydrogen molecules inside the stomach go and react with the chloride of outside the stomach cell and forms HCl. Hence, the secretion of HCl increases inside the stomach. So, if we block the binding of histamine with H2 receptor, we will stop this cycle, and thus the secretion of acid is mediated or it is decreased down. Now, this is H2 antagonistic drugs, which are also called as anti-ulcer agents because they treat ulcers, and since they secrete or they decrease the secretion of acid, they are also called as anti-secretory drugs. They are used for the treatment of acid peptic disorders, heartburn, gastroesophageal reflux disease called as GERD, erosive esophagitis, gastric and duodenal ulcers, gastric acid pathological hypersecretory diseases such as Zollinger-Ellison syndrome. They are also used in combination with H1 antihistaminics for the treatment of chronic urticaria. The drugs which are used in combination is cimetidine, famotidine, and ranitidine. So let us see the structure. To learn and understand the structure of cimetidine, we have to break the structure. The name is broken into two parts. Dine represent guanidine, E represent ethyl, ET represents ethyl chain, and IM represent imidazole ring. So when we talk about the structure of cimetidine, we will see that it consists of a center chain of ethyl. So this is your ethyl group. The first carbon of ethyl is attached with guanidine, methyl cyanoguanidine, and the second carbon is attached with thio group, sulfur, and imidazole, methyl imidazole ring. So the structure is understand by the IUPAC name. Here the nitrogens is numbered as 1, 2, 1, 2, and 3. What is the guanidine? Guanidine is an NH2 C double bond NH2. So one of the H is replaced by cyanide group. Here we will write 1 cyano, 2 methyl, 3 from 3 the second carbon is attached with sulfur, and the second carbon consists of 5 methyl. So 3, 2, 5 methyl, 1H imidazole, 4 yl sulfonyl ethyl guanidine. This is the IUPAC name to understand the structure of cimetidine. Come to the main part of this video lecture series, that is synthesis. To start the synthesis of cimetidine, we have to understand the first chemical compound, the precursor of the reaction. So the precursor or the starting material of this reaction is a imidazole derivative, which consists of methyl group at fifth position, and the fourth position consists of ethyl formate. What is formate? Formate COOH. So H is replaced with ethyl group, so ethyl formate at the fourth position. This precursor reacts with the water, or the first step undergoes hydrolysis reaction. So when the hydrolysis reaction take place, we get a intermediate, that is imidazolic acid. This is your imidazolic acid, methyl 5 methyl imidazolic acid. Now in the second step we will see the reduction. The second step, there will be the reduction reaction. Reduction takes place. So if the reduction takes place, the hydrogen is attached with the carbon, we will get an ethanol group CH2OH. Again, this intermediate 2 reacts with 2 amino, this is your 2 amino ethyl thiol group. This 2 amino ethyl thiol group is reacted with intermediate second. So water molecule is separated and the intermediate third is prepared. So this is your third intermediate, which consists of methyl at 5 position, and at fourth position we will consist of amino ethyl thiol group. This reacts with dimethyl cyano carb imido dithionate. They consist of two sulfur groups, so dithionate. It reacts with the intermediate 3. When the reaction takes place, one of the thiomethyl group is separated out. So in the fourth step reaction, we get a fourth intermediate. When this intermediate undergoes methylation reaction with methylamine, CH3NH2 is your methylamine. The reaction takes place, and again one of the thiomethyl group is separated out. So when one of the thiomethyl group is separated out, remaining NH will go and attach with this carbon atom. When this carbon atom is attached with nitrogen, we will get the main structure of cimetidine, main structure of cimetidine, which consists of 5 methyl imidazole ring attached with a ethyl guanido thiol group. Thanks for watching the video. If you like it, please subscribe our channel, that is Modern Group of Institutions, our YouTube channel MGI.